All right. Okay. So here's where we're going to start today. By the way, we're going to take a little bit of time today to do something we've done several times before, but not for a little while, where we are given a series of observations and we design or we come up with a hypothesis explaining those observations, and we come up with an experiment to test our hypothesis. Okay, but before we get there, we need to review a little bit. So somebody remind me the three parts of how a cell receives communication, right? So communication is the transmission and the reception of signals. Mark.
Right. Chemical, tactile.
Oh, so you're going specific. That's fine. You can go specific types of communication. So
chemical,
tactile.
Electrical? Is that?
Electrical is one that we haven't talked about yet. It's a fifth.
Yeah.
So chemical, visual, tactile, auditory, and then electrical.
Oh, I was just talking about the cells.
Oh, cells.
Yeah, that's besides what you said.
Yeah, no, you're absolutely right. So cells are chemical, tactile, and electrical. Boom. Boom. Okay, so if communication is the transmission and the reception of signals, what are the three parts of how a cell receives a signal? Tori?
Receptive.
Yes, receive the signal. So binding the signal or binding the ligand. Good. Then yeah.
The cell transduces
the signal, which transduce, what is an analogous term for that? For transduce? Yeah, translate. Doesn't mean the same thing, because transduce is a very specific chemical process. And Tori, the third part, which we didn't get a chance. Yeah, the cell changes in response to that signal. Good, wonderful. And so we talked about those first two on Monday. And so what are our different ways that we can receive a signal? There are four of them. Okay, there are four. Yeah, Carrie.
A G-protein coupling receptor.
G-protein coupled receptor, or coupling receptor. Okay, and so in that receptor cannot act on its own, but that receptor activates a G-protein that then carries out some task, right? And how do you activate a G-protein? How do you turn it on? Emma?
If it's bound by GTP, it's on.
Yeah, you've got to phosphorylate that GDP, right? Turn it into GTP, and then that G-protein can take that extra phosphate group and phosphorylate something else. Okay, what's another way we can receive a signal? Katie.
There's the
Receptor tyrosine kinase. And so here's a receptor. It does not have to be coupled to a G protein, because when the receptor binds the ligand, it becomes active. And how do you know that a receptor tyrosine kinase is active? What is present that is not when it's not active? Emma? Yeah, it's phosphorylated, and two individual receptors bind together. Okay, and they're all phosphorylated, and so we've got all of these tyrosine residues phosphorylated, and then they can do what with those phosphate groups? They can phosphorylate something else. Okay? And that's why we call them receptor tyrosine kinases. Because what is a kinase? Something that phosphorylates something else, usually another protein. Okay, so then, so what's the third? Yeah, Emma. An ion channel receptor. And so here, in the presence of some ligand, what does the channel do? It opens up, allowing ions to flow through. And ions can't typically go through the cell membrane. Why? Yeah, Emma. Because they're charged, and you've got this non-polar environment in that phospholipid bilayer that does not interact well with charges. Okay? And what is the fourth way? Yeah, Katie?
Intracellular receptors.
Intracellular receptors. And what types of things can actually get to an intracellular receptor? Yeah, Emma? Yeah, things that can diffuse through the cell membrane. And so they better be small, or they better be nonpolar, or preferably both. Yeah, small and nonpolar. Those are beauties. Okay, so now we've bound the ligand, right? We've bound the ligand. Step one is done, and then what is step two? Emma? Transduce the signal. And there are basically two different chemical pathways that you can use to transduce the signal. What are they? Katie?
Kinase cascade.
A kinase cascade. So you phosphorylate something, you phosphorylate a kinase, protein 1, that does what?
Phosphorylates protein.
Phosphorylates protein 2, which is another kinase, which does what? Phosphorylates a third kinase, ultimately until you phosphorylate whatever that target protein is, that on switch, and you're good to go. But what happens during the cascade in addition to just phosphorylating a series of kinases? What happens to the signal? Does it get louder, quieter? Yeah, it amplifies, it gets louder. Because every time you activate an enzyme, that enzyme can activate a whole bunch of protein 2s, right? That protein 1 can activate a whole bunch of protein 2s because the protein 1 doesn't get used up in the reaction that activates protein 2, right? Because it's an enzyme. And enzymes aren't used up by the reaction. Okay, what's the other one? If it's not a kinase cascade trinity.
Secondary messenger.
Secondary messenger. So rather than ushering a kinase cascade, you might, at that receptor that binds the ligand, might instead either lead to the production or the release of some secondary messenger. And our two most common secondary messengers are, Rick? Cyclic AMP.
Oh yeah, I have that right there.
Cyclic AMP.
But that's like A.
And calcium ions. Okay, I want to real quickly, so that's all of our review, but I want to real quickly show you what cyclic AMP is. Okay? I want to real quickly show you what cyclic AMP is. Alright, you all will remember this from our discussion on biomolecules, a nucleotide. So a nucleotide has a sugar, and most of our nucleotides, the sugar is ribose. And then over here it's attached to some nitrogenous base. And what bases might be there? Well, they're nitrogenous, so they contain nitrogen. But this is like the ATCG that you all learned in Honors Bio, right? When you talk about DNA and you've got A, T, C, and G, right? Adenine, cytosine, thymine, guanine. It's coming back. This is like riding a bike. Except for this bike is awesome, right? It's like riding a bike and how it comes back to you, and then you get back on the bike and you're like, wow, this isn't as much fun as I remembered it was, right? But this is the kind of bike that when you get on this bike, you're like, man, this is everything I imagined. This is the most fulfilling thing I've ever done in my whole life. Okay. All right. So everywhere there's a vertex on this, what is this? This is a pentagon. Okay, everywhere there's a vertex, there's a carbon, unless we show that it's something else. Like at this vertex here, it's oxygen. But so this is carbon 1, and this is carbon 2, and this is carbon 3, and this marker's dying. And this is carbon 4, and then there's carbon 5 right here. Okay? So how many carbons are in the sugar ribose? 5. How many carbons are in the sugar glucose? 6. So this sugar's a little bit different, right?
What is this?
This is ribose. But this is 2 parts of a nucleotide. Okay? Now attached to carbon 5 on a nucleotide. Oh man, my 5's in the way. I'm gonna move it down here. Is a phosphate group.
Do you remember when we did that whole big thing, Katie? Yeah.
Wait a minute.
Wait a minute.
Yeah. No. See, that was easy, low-key, though.
What's oh yeah.
Sorry, got a little carried away there.
Is this still chapter 11?
Yeah.
Wait, do you
Yeah.
want with it down?
Wait, is this cyclic AMP?
Well, not yet. Right now, what would make this AMP? Our base would need to be something specific. Which base starts with an A?
Oh, see,
Adenine.
calcium.
So if this is adenine, this would now be AMP because how many phosphate groups does it have? 1. And what does the M in AMP stand for? Mono. What would make this ATP? If we added more phosphate groups.
I had one.
Ye-
That is a very different. It's one of the most contagious things that there is. So there are way more ways to get it than kissing someone.
You can't just like drink from a water bottle.
Okay.
Yeah, that's
Now,
that's
shh. Let's focus. Remember,
Dang, that's a lot of
Put-
copies.
put away the background noise. Okay. So, hey, this is now ATP because it has how many phosphate groups? 3. And what does the T stand for in ATP? Triphosphate. Okay. I'm just showing you this. I think we've drawn this before.
Yes, we have. Yes.
But this is not cyclic AMP. 1, there are 3 phosphates on it, so this is ATP, right? So in order to be cyclic AMP, we've got to get rid of 2 of these phosphates. Okay? But that's easy to do. We get rid of phosphates off of ATP all the time, right?
Yeah.
Every time you take an ATP and you convert it into ADP, you remove a phosphate group, right? So look, let's do that. Boom! We removed that phosphate group, released energy, fueled something we wanted to fuel, and now we have ADP, right? But we don't want ADP, what do we want? We want AMP, yeah, Trinity.
Is there a third one on there, or did you just read it?
There was a third one that I just erased because I released energy. Did you feel it? Yeah, I released it. Okay? So we're gonna... Okay, so we're gonna make AMP, and we're gonna break another phosphate group off, and what's gonna happen? We released energy. Did you feel it? Yeah. So now we have AMP, but this. No, I got the list of people that were going for choral ensemble, and none of you were on the list. I checked.
I'm definitely on the
They sent out a list on Monday and
They take me off.
said, please release these people for choral ensemble, and I checked the list.
Yeah, they must
There.
have taken
It must be some kind of mistake,
I
right?
don't know why they do that, Dr.
Okay.
Engel, because somebody pretended to be on the golf team for the entire season, and they got released at like noon every day, and they just dip every day.
Okay.
Aaliyah.
Okay, but this is not cyclic AMP until something happens. This bond has to get bent around, so this phosphate group bends around. There's still carbon 5, now I'm going to move my 5 back to where it was. Look at that! Look what we did here!
Why would we ever do that?
We made another ring, and this is why it's called cyclic AMP. Why would you do that? Because this molecule here can trigger several different chemical reactions.
You don't need that carbon.
No, you didn't add anything. You just reoriented the phosphate group to facilitate this oxygen picking up a bond with carbon 3.
That's
Yeah.
nifty.
How does that happen? How does it benefit?
It has to get physically. I don't know that this is the case, but I think this is what I remember because I didn't look it up. I think what you actually have to do is remove that phosphate group and reattach it a different way. And so you have to have an enzyme that'll do that.
Yeah, like this. Adenylil, whatever it's called.
No, adenylyl, yeah, adenylyl, yes, adenylyl is what makes, I said no, but yes, adenylyl is the enzyme that makes cyclic AMP. Alright, adenylyl. And I didn't write it down again, so I don't remember exactly how to spell it. I think it's ADENYLYL.
That's correct.
Adenylyl.
ADNY.
Okay? So isn't this cool? This is our secondary measure. It is cool.
I know, but I can't keep track.
This is our secondary messenger, cyclic AMP.
That like
3 was
ATP. And you get that this has to be AMP because it's adenosine, which is right here, and then there's only one phosphate group. Okay?
No.
Alright, so now we're all reviewed. And the only thing we need to do is now do step 3 of the cellular response.
Wait, what?
Right? Well, communication is the transmission and reception of signals.
How many steps is it?
Well, it's just one. It's 3, and there are 3 different things that usually
The first
happen.
step. We already did 1 too.
Yeah, I know, but how many like sub-steps are in?
There are only 3 that we'll talk about. All right. Okay.
Good question.
May I erase this?
No.
But our secondary messenger is activating a lot of reactions that we need to stop. So I need to erase our secondary messenger.
Do it.
Okay.
Stop the reaction.
All right.
Now it would never happen.
So we're all done with our review. Yeah.
So the adenines make the AMP.
The adenylyl is an enzyme.
Oh, okay, and then that's why it makes.
So then a little AMPs.
Yes. Because you can't just make. Hey, we can't just make cyclic AMP by removing 2 phosphate groups. That makes AMP. That does not make cyclic AMP. To make cyclic AMP, you've got to put that phosphate group on a different way so that it binds to 2 different carbons instead of just 1. Yeah.
Okay, I understand the difference between cyclic and like the normal one, but like, what does it do? That is different from.
What does cyclic AMP do?
Different
It's
from normal.
that's a good question. It acts as a machine that can activate other processes. What it actually does varies a great deal depending on what it's activating. If you go back to our work on enzymes, a lot of times you have what are called allosteric regulators that bind to a separate place on that enzyme and change the shape. Sometimes that's what cyclic AMP does. Okay? Does that make sense? Kyle.
And then you have to have.
Man, I wasn't prepared for you guys to ask so many questions about cyclic AMP. I should have been more prepared and read more about it to remind myself. Going beyond the text. Yeah.
You have to have 3 phosphate groups.
For ATP.
But once you break those 2 off, then you can okay. So what I guess I'm trying to ask is, can you just take the 1 phosphate group and then put it down, or like?
Well, adenylyl can, but you can't. But adenylyl is inside of your cells, so I guess you can, and you do.
So like you don't need to pick the 2 other phosphate groups.
Well, you have to have just AMP in order to make cyclic AMP. Yeah. All right. So remember, step 3 is the cell changes in response to binding and transducing that ligand. Okay? And there are lots and lots of things that a cell can do, but we're only going to talk about 3 of them. 1 is that the cell can grow in response to that signal. And this is like all of chapter 12 is 3. I am so okay.
He's jazzed.
I am jazzed. Okay? So all of chapter 12 is about this one, that the cell can grow. And we'll say grow / develop. Okay? So that's one thing the cell can do. And if you're like, what does it need to do that? That's what today's all about. That is what chapter 12 is all about. Okay. Letter B. What, is it misspelled?
No.
Okay.
I don't know how to spell. That's what I was asking.
So letter B is it can transcribe some new product. And this is what 2 weeks from now is going to be all about. So if you're like, Dr. Engel, like, what's that all about? Don't you worry, because we'll get there in a couple of weeks and we'll think back to this 3A, and this is a cell's response to some signal. It is the end of the communication, right? Okay. And then step 3, or the third option that we're going to talk about, is apoptosis. Apoptosis.
Thank you.
They're coming. I was going to throw it to you. Okay? The last one is apoptosis. And apoptosis, there are 2 main things that have to happen. Degrade cell contents degrade cell contents, and then I'm going to put this in quotation marks because it's not actually an explosion, but it's kind of an explosion. So degrade cell contents, explode the cell.
Doesn't that kill it?
Yep.
And why would you want it to happen? Like for the bad cells?
Is that all in the parentheses?
They're
See, that's like
Yeah, is that all in
covered
parentheses?
It's very ambiguous.
So there are two main things that happen in apoptosis. You degrade cell contents, some of those can be recycled, and you explode the cell. And absolutely, this leads to this cell death. There are three main reasons why you might do apoptosis, but Issa, your question? Oh, so you want to go on a bad cell? Yeah. So there are three reasons you might give a cell an apoptosis signal that will trigger this. One is if something's wrong with that cell. And so what might be a clue that something's wrong with the cell? Okay, that there's some mutation, that cell is not behaving properly, which is an indication that there's some that there's something wrong with the program, right? If the cell's not behaving properly, it oftentimes can be because there's something wrong with the programming. There's something wrong with the DNA. Okay? And so that might be why you give a cell an apoptosis signal. Emma, are you going to give us another reason? Okay. Another reason is that cell is infected. Okay, if the cell is infected, were you going to say that?
Say it's sick.
Oh, man, nice. I'm so sorry, Kyle. Yeah, if that cell's infected with something and a white blood cell can recognize that, it might trigger the or it might just eat the cell and then just tear it apart. Emma, your question?
Some like bad cells disguise themselves as good cells.
Yes.
Then how can
Undercover.
yeah, like how do you know if it's like do they just not get killed or
Correct. Yeah, Trinity. Trinity. Okay.
Oh yeah. So do they? Never mind.
Okay. So two other reasons why you might trigger this process, other than that there's something wrong with the cell, is there are too many cells. Okay, there might be too many cells, and so it is a means of population control. Okay, and there's something wrong with the cell. There's actually another one. It could be malfunctioning, it could be infected, or it could be just old. Like that, their cells have like a certain length. And so, but yeah, also there might be too many cells, and the third reason is it might be necessary developmentally. For instance, when we develop, have any of you ever seen an ultrasound of a baby that's like maybe 3 months old or so? And you can see their hands and they've got webbed fingers.
Really?
Yeah. So there's skin that connects all of the digits. And so what erases that webbing is apoptosis in those cells.
Wow.
Yeah.
so if it doesn't happen, people will just have like
Correct, and that's how some people are born with webbed digits.
Okay, Emma.
Okay, well Emma first because she had her hand up and then Rich.
why doesn't this work for cancer cells?
We'll talk about cancer cells today.
They're evil.
Yeah. The main reason, I'll answer it and then we'll talk about it more today. The main reason this doesn't work for cancer cells is because cancer cells shut themselves off from external signals. They lose the machinery. I mean, they still receive it. They'll still receive the ligand or bind the ligand, and then they may or may not transduce the ligand, but they don't change in response to it, even if they transduce it, because the machinery that would trigger their change, the machinery that would trigger them to go through apoptosis or trigger them to, respond in a way by transcribing some new product, that machinery is malfunctioned. And then eventually they'll separate from the surrounding cells and they'll basically just shut themselves off to all signals. Alright, Rick and then Michelle.
Just kind of like an Emma level question.
Yeah, you already said
I
that.
heard somewhere that's like, if you have a baby and it's like right after it's born, like if you put it in water, it would know how to swim. Is that
I don't know if that's true, but so when babies are first born, they don't breathe, right? Because even if they've been practicing breathing in utero, the trauma associated with the birth process, they'll stop breathing. And so if a baby is born in water, a lot of times they'll do a water birth. The baby's not going to drown because it's just not breathing at that moment. So it's not going to breathe in a bunch of fluid.
When it comes out.
Well, it's attached to the umbilical cord. That's where it's getting its oxygen. And then you actually typically you have to induce a baby to take its first breath because it won't do that. And so there are a number of ways that you can do that to get that baby to take its first breath. But once it takes its first breath, it's dialed in, right? That's how it's going to get its oxygen now. So my guess would be what you heard was associated with like that water births are possible, but it's not because the baby knows how to swim. It's because it doesn't breathe underwater. It's still attached to the umbilical cord.
Wait a minute.
Because I'll tell you this, the baby has spent, if it's human or a cow, the baby has spent 40 weeks in a warm, the time is the same. The baby has spent 40 weeks in a warm fluid, and that's the environment that it knows. The baby's not going to choose to leave that environment. So it's not going to like swim to the surface to get out of that environment. It loves that environment. So if you do a water birth, they do it in like a warm body temperature pool of water. Okay, Michelle and then Emma. Transcribe is to write. Alright, Emma.
Oh yeah. That was like weird. Like, what happens when your water breaks? Like what causes that?
Yeah. Basically the wall of the placenta breaks down, starts to break down towards the end, and so it'll release the amniotic fluid that's inside. So that's what's happening is you're leaking amniotic fluid, and that's why once your water breaks, they want you to deliver pretty quickly because you've either lost all of or most of the amniotic fluid, and it's no longer safe for the baby to be in there. Okay, and so once your water breaks, so even if your water breaks at like 32, 33 weeks, usually they'll induce labor because it becomes very challenging to try to. Now, if it's really early in your water breaks, they might try to repair the placenta and hope that you'll make more amniotic fluid. It's not that simple. Yeah.
Tap water in here.
Alright. Isa.
Wait, so the baby, you said the baby's not breathing?
Correct.
So then how is it floating?
How is it?
Like, it's like, hold on, look. Let me give you guys the facts. I was in my pool one day, just being curious. And I was like, I think if I breathe all the way out and just float, I'll go down. And I did.
Yeah.
So,
Was
maybe go down. like if I like this, and I went underwater and I float up. I went, and then I float down.
Yeah, babies' bodies are a little bit different, and they have a lot of gas in their digestive
So
tract.
that's keeping them up.
Probably. Yeah, but I don't know the buoyancy constant of a baby. So, all right. Any other questions? Okay, here's what I need you to do. Listen up. Listen up, everyone.
I have a question.
Yeah, Tori.
Can a baby rupture one of your internal organs for kicking?
Yes, absolutely. Well, cause some kind of a bleed. Yeah.
Does it happen?
No. No, because there's a cushion. The amniotic fluid provides a cushion. So, all right. So I need you to write down these observations. Okay. Observation 1. Most cells.
Is this chapter 12 or chapter 1?
What is that? What?
This is the bridge between the two.
What is the title of this?
Are you upset because this isn't capitalized?
No.
Is that the problem?
I'm sad because I don't know.
We don't know what this is.
I told you. I told you all that today I was going to give you a series of observations. You were going to take those observations. You were going to come with a hypothesis to explain those observations. You were going to come up with an experiment to test your hypothesis. And then something new, you're going to come up with a way in which you would model those data. Okay, just relax. And so you're like, if this is chapter 11 or chapter 12, The answer is yes.
So which one?
Both.
Oh my God.
So put it in either one. Put it in either one. Okay. Most cells do not divide.
Don't you think I want to look at page?
Okay, most cells do not divide, and this is talking about like most of the cells presently in your body do not divide. Number 2.
Let's sound like me.
Cells. Except for that was not genuine. I'm sure your coughs are genuine.
Oh, mine are-
Mine was a means by which to say, "Would you please
Stop
be
talking.
quiet?" Because it was either that or grab an imaginary telephone and call you from over here.
I would have liked that. That would have been funny.
Isn't that what Mr. Lee does? Although he has an actual telephone, but it's not attached to anything.
You did it in my class.
Cells
Somebody just sprays
require signals to begin. Whoa! Begin dividing. Okay? Cells require signals to begin dividing. Okay, so most cells do not divide, and the cells require some signal to begin dividing. They have to be told to divide. Okay?
So what if they don't get the signal and they just? They don't
Well, we haven't even gotten the third observation yet. Cancer cells divide in any environment. Okay? So most cells do not divide. Cells require signals to begin dividing. Cancer cells will divide in any environment. So what I want you to do is note these 3 observations, and I want you to predict what is different about cancer cells as compared to normal cells. Okay, Mark and then Emma.
So you're saying if we just yeet cancer cells into space, they'll still be divided?
Well, I mean, they would die in the vacuum of space. Record. We are now.
Can you turn the air up, like warm?
It's already, the thermostat. Okay, the thermostat is set to 70,
Okay.
and it is currently 69, so it is working its way up to 70.
No?
Here, I'll set that at 72.
Yeah.
Okay, alright. So we're transitioning to chapter 12. Chapter 11 was called cell communication. Chapter 12 is called cell cycle.
Wait, this is chapter 12?
This is chapter 12.
Oh my gosh.
I don't understand. Okay, so chapter 12. As we begin chapter 12, I have 2 questions for us to answer. And as we end our discussion of chapter 12, I have 1 question to end it. Okay? So the first question, why divide? So chapter 12 is about cell cycle, and it's all about dividing and making more copies of yourself. Okay? And so the first question we have is why divide? And there are basically 3 processes that require cell division, cell multiplication. Okay? Trinity.
Did you say the name of chapter?
Cell cycle. I only said it 3 times.
I think you should just say it once just to be.
I think everybody good?
Oh, this
You know what I could do is not even audibly say it, just mouth it. Like chapter 12 is called.
I could hear it.
Yeah.
I can't do it without even.
You don't hear when it's just annoying anyway.
We play this game at the dinner table to get our kids to not talk so loud. Where we'll have them mouth a word and then everybody has to try to guess what they've mouthed. And my son, he's 5, he is amazing at it. He is so good. Like I can't do it without making any sound. I have to make some sound if I'm gonna get my mouth to go that way. And our oldest is like that. I have a hard time, but my son, he is so good. And you can see, I mean, he's so careful. He's like enunciating without making any words to press. Okay, so why divide? They go to school.
We can take them out one day.
they're on spring break a different week than we are.
Wait, why are you that?
That's just so fun.
I might. Okay. They can't go. My kids can't go here for free. It's only full time. Yeah. Anyways, okay. So why divide? Why divide? There are 3 processes that require cell division. One is growth. So the way we grow is by dividing and multiplying cells.
Wait, I thought most don't even divide.
Well, when you're an adult.
Wait, what?
But when you're growing and developing, everything is. I mean, if your bones are gonna get longer, your muscles are gonna get more developed. Okay, so why divide? One is growth. Another one is development.
So you see space between these two ones?
No. And then the third one. Somebody tell me what the third one is. And so the first 2 don't happen once you reach adulthood. If you're a mammal, if you're a reptile, you keep growing for the rest of your life.
Reproduction.
What's that?
Repair.
Where did you get that?
My head.
I said growth and development. He's not that special.
Dude, Kyle, you've been killing it today with the cells are sick.
That wasn't it.
No, and then repair. What are you talking about? That's one thing. That's 2 things.
Yeah, that's good.
And has anybody else in here had 2 things today?
Now, Katie's my witness. I said growth and
And the
development.
Okay, we get it right.
The third one is repair. And this is the one that happens throughout adulthood. Growth continues in some tissues, like again at the base of your skin. That will continue throughout your life until it stops, and then you get wrinkly and your hair starts to not regrow. So when you shed it, you just don't get it back. Right? So you're like, no. Every time you brush your hair and you find some in the brush, you're like, no, because it's not coming back. But then you get a smoking wig. Just a smoking wig, and you rock that. You rock that.
Your hair starts getting gray, but stops.
Oh yeah, you lose the ability to sequester pigment into the hair. So the individual hairs are actually they're either white or clear
So what?
Okay.
when you start to go gray,
Wait, clear.
Clear hair?
Clear hair.
like a polar bear.
Clear hair.
Yeah, polar bear's fur is clear.
That's why.
They do. They have black skin and clear hair.
So how come
So how come their hair looks white?
Because the clear hair reflects a lot of light back, and the light is white.
So what happens when we
shines
take
bright
away
like
the
diamonds.
What if they go in the shade?
Oh, it's still the same because it's just reflecting white light.
So if you cut hair,
Wait, what if they go in the shade? They're still white.
What if they go in the shade?
Hey, what if you put them in the dark?
Well, you can still see in the shade, right? There's a small amount of light.
So it's still white. It's still white.
Yeah.
What if you put them in? It's supposed to.
So you're talking about like if it's like real dark, you know, like when it's
No, you
real.
put them in like a dark closet, absolutely no light.
Well, you can't see anything like that. Oh, and a flashlight, it's gonna reflect back the white light.
Wait, when do they look black?
Those of you listening to this recording right now, you realize that I've completely lost control of this classroom. If nobody ever finds me and this is the last record of my existence, just know that I'm afraid
When do they look black?
and very, very disappointed.
What did I just miss?
Emma.
When do they look black? Yes.
Okay, alright, no more just, okay, shh. If you shaved a polar bear, it would be black, because that's what color their skin. Okay, listen, no more talk of polar bears.
I can't believe their hair is.
Okay, so these are the 3 reasons why you need cells to be going through the cell cycle. Now, we know that cells won't divide unless they receive a signal to divide, right?
Yeah.
Okay, so these are great reasons for a cell to divide, but they will not divide unless they receive a signal to divide. And there are 2 signals, just a moment, Emma. There are 2 signals. So these are signals to divide. Signals to divide.
It's gonna be long.
All three
No.
of them are?
No. These are the reasons to divide. These are signals to divide. One is called growth factors. Growth factors. And the other one is an anchorage signal. Growth factors and an anchorage signal. What does it mean to be anchored to something?
Like you're stuck.
You are stuck, right? Which is such a wonderful name for the capital of Alaska. Anchorage. You are stuck.
Juneau,
Juneau is the capital, the largest city in Alaska. Nice call. Man, I usually nail my state capitals, but yes, Juneau. Juneau, I think, is the smallest. Is Juneau the smallest capital in the United States? It's either that or Cheyenne.
What province?
Yeah,
It's
that's
not
not
Providence.
Providence.
It's Austin.
Providence, Rhode Island, in numbers of people? No way.
Phoenix is pretty big.
Juneau's got. There are a lot of people that live in Rhode Island. The state is small, but Juneau's got less than 50,000 people.
Montana.
Cheyenne.
Is there?
What's the capital of Montana? Helena?
Yeah.
Yeah, Helena's pretty small too. And then Bismarck, North Dakota. Okay, shh. Emma, did you have your question?
Yes. So when people are really old and they start shrinking, does that have anything to do with cells or does it
So reduction in bone density. You're losing the extracellular matrix in bone. Okay, shh, guys. Okay, so an anchorage signal, the cell has to be physically attached to the extracellular matrix to grow, or at least to some other cell. So it needs to be physically attached and it needs to receive these growth factors, unless it's what kind of cell?
Cancer cell.
Cancer cell. Cancer cells no longer need growth factors.
Those are the exceptions to like all.
Wait,
Yeah.
there's only 2 signals?
Yeah, growth factors and anchorage signals. Okay, yeah, Trinity.
Which one has to be physically attached?
Which one? All of the cells.
No. You said has to be like physically
which factor?
Like current cancers or
Wait, I'm confused. So for any of these three reasons are why it'll grow, but the cell will only grow if it receives a growth factor, a signal to grow, and it's physically attached to other cells or to the matrix.
You say like, are both signals have to be physically attached or separate?
Both signals need to be present for a cell to grow. It has to be receiving growth factors telling it to grow, and it has to be physically attached to other cells or to the matrix. Okay, shh. Give me 15 more minutes. 15 more minutes of focus. Okay. What happened?
Because your divide looks like divide.
Oh, okay. Alright. Okay, this is disappearing. Okay, so this is Y divide. The next question is The next question is what divides?
Cells. Yeah.
Well, it is the cell absolutely divides, but it's also
The genes.
the chromosomes.
How many chromosomes do we have?
46.
I was gonna say
23 pairs.
I thought
Yeah.
they were, I thought it was 23.
23 pairs. One from mom and one from dad. So for chromosome 1, one of your chromosome 1s come from mom, one of them came from dad.
So then when you have kids, you lose chromosomes. One
What?
No, because every cell in your body has 2 cells.
The new ones are
Yeah, Emma.
Okay, so like,
Guys, I have
when a
had a
child is lacking one chromosome, they have Down syndrome, right?
No, it's an extra copy of chromosome 21.
Oh, so like, what's the new? What is
It leads to overexpression of genes that lead to developmental abnormalities
Where are the chromosomes in the body?
in the nucleus of every cell.
What?
Okay, listen.
I didn't take bio, so I don't know.
Okay, the cell needs to divide. In order for the cell to divide, it also needs to divide the chromosomes evenly between the 2 cells produced as the cell divides. Meaning, prior to the chromosomes dividing, what has to happen?
Organelles?
They must be duplicated.
Well, they're gonna have to divide too.
That's true, you're right. Must be duplicated before division. Because if you're going to divide the chromosomes evenly between two cells, you are going to lose half of your chromosomes, right? Unless you duplicate them beforehand.
That's like telephone.
Telekinesis.
You're talking about
Telophase.
telophase. Telophase is the very end. Okay. So the chromosomes must be duplicated before the division, and the two copies are physically attached together.
Like stapled together.
Yeah, sort of. They're attached by a protein
It's like-
called.
What's that paper called?
The
It's like
protein is called
together,
securin.
It goes on both and you can like rip it
Oh,
apart.
those are
Transfer
cool.
paper.
Yeah, it's like that.
Okay. So the two copies are physically attached, and we call these sister.
Haxa.
No, sister.
Chromosomes.
Sister copies.
Not chromosomes, but that's close. Sister chromatid.
Oh, yeah.
Okay, so the DNA must be completely duplicated. You've got to copy all of the DNA in the nucleus of your cell. Okay, you have 46 chromosomes. You have to duplicate all 46 chromosomes. The two copies are physically attached together as sister chromatids, and this is what it looks like.
Like an X.
Like an X.
Not everyone.
Okay? That's what it looks like. We've got, here's one sister chromatid, here's the other sister chromatid.
It's like they're like that. It's not an X, it's like that.
Okay?
Does that make sense?
It's like this.
It's like this.
Sometimes it's like this,
Fingers backwards. So
sometimes it's like this, sometimes it's like this.
I got you.
I was like, not
joined in the middle.
yeah, there it is. It's like that.
They're not always joined right in the middle, sometimes they're joined up at top.
So they're not crossing. They're like
No, they're not crossing, they're side by side, but they're only connected at one point. Okay, alright. Does this make sense? Okay, so in order to divide the chromosomes, and Rick, you are absolutely right, you also need to divide the organelles. Those have to divide as well, but we don't talk a lot about those when we're talking about cell
that
division.
just kind of happens.
It doesn't just happen, but
It's not
yes,
important.
it is important. We
Well,
just
let's not talk about it.
because it just makes it way more complicated.
Doesn't that kind of go through the same process?
Depends on the organelle. Yeah, Michelle, did you have a question? Okay, all right. Any questions here? So you know what divides. Okay, you know why we would divide cells. Now, yeah, we can do how, and we're gonna do mitosis. Okay,
Hey.
so I want to draw, and I'm gonna draw a pie chart for you.
Oh my god.
Oh no. You're gone.
You should revoke your PhD.
But here we're not comparing two groups. We're looking at a single cell, a single cell, a single cell.
She's single.
Oh, 6 o'clock.
That's not even 6 o'clock.
Oh, I know exactly what chart.
Okay, so this circle represents the cell cycle.
Yeah, that's not that good.
Oh, wait, so what should we label this
We
like?
used to do
Section
this.
You should label this circle cell cycle. Sure. Okay. This portion of the cell cycle is called G1,
G2.
which stands for first gap. All right? This portion of the circle is what?
G2.
Nope, G2 is here.
You thought.
And that's second gap.
G 1.5.
No, wait, it's got an actual. I don't
Hey,
think
What's the name?
Telophase.
S
Quit
phase.
saying telophase.
And this is duplication.
Telophase is in S phase, so
During S phase, the DNA is duplicated. There are also some other machines duplicated that are necessary for carrying out the cell cycle. Okay? All of this together. All of this together. All of this collectively is called interphase. Okay? Do we got that?
Yeah.
Okay, all of this together
What's that little?
is called interphase.
Oh, that's the. Okay, never mind. I'm good.
You're like, what is that other sliver?
We're probably about to go over that right now.
We are.
What's that thing around it? No, not the sliver.
This whole thing,
Yeah.
it's just to show us that everything from here all the way to here is interphase. This right here
Me too.
is called M phase. Okay, that right there is called M phase. M phase. What do you think M stands for?
Mitosis.
That is part of it. And so this is mitosis plus cytokinesis.
Oh, that's just yeah.
Wait, does that say S phase duplication?
Yep. So M phase is mitosis plus cytokinesis. It looks beautiful.
No, I'm talking about mine.
Yours.
Wait, that says
Everything is beautiful.
I hate it.
Everything is beautiful in its own way.
Yeah,
Everything else in my
Where
notes
does it-
looks fine.
You know, the only thing that makes us beautiful is that we were created by God in His image. So I'll tell you what.
What's in
That's the only thing beautiful about me. What's up? And this dimple. Yeah, Trinity.
Dimples are so cool.
what's in the sliver? I can't read it.
Mitosis plus cytokinesis.
I had like 1.5.
Yeah, I had a shadow once.
Okay, so we've got interphase and M phase. Interphase is G1, S, and G2. This sliver is M phase, which is mitosis and cytokinesis. Okay?
Cytokinesis is just like the transition thing.
Cytokinesis is splitting the cell into two.
Oh yeah.
Mitosis is splitting the genetic material in two. Okay? So mitosis, this doesn't have to be off to the right of your circle. It can be beneath your circle. Or under your circle. I guess that's what beneath means. It can be on top of your circle. It can be over to the left of your circle. It can be on a completely separate page even. Okay? Mitosis. Mitosis is nuclear division. Mitosis is nuclear division. Division of the nucleus into two nuclei. Right? Because we already knew that we had to divide the chromosomes. Mitosis accomplishes that. Emma.
So is that entire phase?
Interphase?
Yeah.
From here all the way to here is interphase.
So just not including M phase?
Correct. Yep. And then cytokinesis, which is also drawn in that little sliver, is cellular division.
Cytokinesis is a weird word.
It is.
I don't like how it's spelled. There's I's where there should be E's.
I spelled it the way
I know that's
Okay.
good. I spelled it.
I did.
Mitosis,
Huh?
nuclear division,
No.
cytokinesis, cellular division. Because we already said, when I put what needs to divide, how did we answer?
The
The cell needs to divide, and the chromosomes need to divide. So, what process gets our cell to divide? Cytokinesis. What process gets our chromosomes to divide?
Mitosis.
Mitosis.
Oh, okay, so this is this.
That's beautiful.
It's beautiful.
It's a beautiful thing.
Okay?
Katie, that is so good. How could you ever say that's bad?
Okay. Yeah, Emma.
Can we cell divide halfway and stop?
Yes.
Listen, plan.
Then what happens to it?
It varies, but ultimately, if it doesn't, it depends on where it stops. If it stops at the beginning, it enters into what's called G0, and it leaves the cell cycle. Maybe never to divide again, which is the way most of our cells are.
Okay, what?
Okay. All right. So mitosis. 5 steps. Okay, I'm gonna leave this up. Okay. Mitosis.
Wait, what's the interface part?
It's
Literally everything but
Yeah, it's just like the cell living its life, right?
Kyle, you've been trying to get us these phases of mitosis
yeah, I can.
all day. Do you remember? So we'll go with telophase, right? That was one. That's the very last.
That's what
Telophase.
Metaphase.
Anaphase is right before that. I heard that from Mr. Garwick.
Metaphase.
Metaphase.
And then I heard metaphase, which happens to be right before it. Man, we are going almost perfectly backwards.
Don't mess it up.
Interface.
What's the other one?
Prophase.
Prophase.
Metaphase.
Prometaphase, then prophase.
Then prophase.
Is that all of them?
That is. Okay? So mitosis has 5 steps. Starting with prophase and ending with telophase. Starting with prophase and ending with telophase. Starting with what? And ending with. Okay. And then we can follow the way we know these stages is there are certain hallmarks that happen in each stage. Okay? Alright. We will pick up here on Tuesday and we will finish chapter 12.